So as Sarah said, my PhD was very eventful in general. So I joined in 2012. And one thing that I would like to show you before I'm going to the science is I looked through my pictures, and I wanted just to kind of show how far we went from the early days, like 2012, 13 of the single cell sequencing to now. You have like a little picture of my lab book from December 2012 when we were trying to sequence 5 cells.
เพราะว่ามันเป็นเรื่องที่เกี่ยวข้องกับสิ่ง
Oh, someone, I can hear somebody.
Jose, I think you need to mute.
Yeah, so I think this is quite amazing, and the pictures I also picked because you can see kind of happiness and excitement.
Pon, I think you need to mute. Pon, I think you need to mute yourself. If you mute yourself, then we can hear Ola more clearly. Pon, thanks. That's awesome. Thank you.
Okay. So I think these 2 pictures I picked because, well, they were also very early days, and they show the excitement that that kind of was around during that time. And I don't know, I had the feeling that we are doing something special, and it was really great because of that we had the chance to do it, which also was not like planned for my PhD, because I was supposed to do something completely different, and then this just landed on my lap, and this made me feel very special, and I'm really grateful for that. And also, I think I chose the Sarah's lab because when I was applying for the PhD, and I was looking for a lab where I could learn computational side of things, and for Sarah that was obvious, and that I can do it, that it's not a problem, and she really believed in me, and that was really also very helpful, and I'm also very grateful for that. And what I would like to show you today is I moved on to the Weizmann Institute, to Elina's lab for my postdoc, where I'm looking into the interactions between host and microbiome. And in general, I'm interested in how external things, so environment and microbiome, is kind of a surface between human and environment, how they influence what happens in the human body. And to look into that, we decided to study acute liver failure, which is a really horrible disease that is in human most often result of overdose of paracetamol, and then we did single cell in mouse model of that, and what you can already see is that we observed a lot of differences between cell types, and when we quantify it a little bit more, you can see that acute liver failure causes infiltration of cell types, of monocytes and neutrophils to the liver, and activation of other cell types, stellate cells, endothelial cells, and Kupffer cells, which are resident cells of the liver. And these activation signatures that happen seem to be quite similar when I looked at them. And then I found that there's a huge overlap between genes that become upregulated in all of these populations when they become activated. And there's as many as 77 genes that are not expressed or lowly expressed in the quiescent versions, and then they become activated and expressed in the activated versions of these cells in the liver disease. And the fact that these are similar suggests that there is a common regulatory mechanism in all of these cell types that drives that, and likely also a common kind of external signal that drives that in all of these cell types. And to identify what could that be, I looked at the promoters of these 77 genes, and I found that in the promoters there's a highly enriched binding site for MYC transcription factor in these 77 genes. So to see if this is indeed a regulator, I used MYC inhibitor, and in mice we performed this disease model using MYC inhibitor to see if we can stop this response, and the readout that we use is infiltration of monocytes, because monocytes infiltrate into the liver through CCL2 chemokine. This chemokine is one of the 77 genes. So the logic was that if we stop MYC transcription factor, there will be no CCL2, and that will lead to no infiltration of monocytes. And indeed, what you can see here, if we give MYC inhibitor, there's no infiltration of monocytes. But on top of that, there's also less tissue damage, which we can see in histology, but also when we look at the liver enzymes in the blood, which is a common measure of acute liver of liver damage, basically. And when we do single cell sequencing in the presence of MYC inhibitor, we find in 2 different models that there's new population arising, which is different than the activated population in absence of MYC inhibitor, but also is different than the quiescent population that is in healthy animals. And this happens in all of the populations that are resident, so stellate cells, endothelial cells, and Kupffer cells. When we look at the expression of the signature, it appears that these genes, again, they are not expressed in the quiescent healthy mice, then become activated in the disease. But if we give MYC inhibitor, they are not there anymore, proving that indeed MYC is a transcriptional regulator for most of these genes in the signature, and especially for the CCL2, which is a driver of the infiltration of monocytes. And in the single cell data, we can also see this abrogation of the monocyte infiltration because of CCL2 that's only expressed in activated cells, but not in the presence of MYC inhibitor. But on the other hand, neutrophils that are also infiltrating in the disease are not affected by the MYC inhibitor. And this is because they are infiltrating through CXCL2, which is not affected by inhibition of MYC. So we can see kind of specific effects of MYC inhibition, and it's not a kind of global effect. So neutrophils are not affected. And when we go to the microbiome, and I performed these single cell experiments also in mice that are germ-free, so mice that don't have microbiome at all. They are basically sterile throughout their life. And I inflict this acute liver failure in these mice. We can see that there is, first of all, less of serum liver enzymes, which are the measure of liver failure. And also there is a higher expression of the 77 gene signatures of the MYC signature in mice that are SPF, so normal colonized mice, compared to mice which are germ-free. And there's the same pattern in antibiotics-treated mice that also are devoid of microbiome or heavily depleted microbiome. And this led me to hypothesis that microbiome acts on the MYC through TLR signaling, and this would be in a way that damage-associated molecular patterns that are originating from damaged hepatocytes in disease normally would signal through TLR to MYC, and the signal would be added to the signal from pathogen-associated molecular patterns from microbiome, and that's why in mice that are devoid of microbiome, the MYC activation is a bit smaller than in mice that have microbiome. And TLR signaling is here the best candidate. And to check if indeed this signaling plays a role in this activation, we used mice that are knockout for MyD88 TRIF. And this is because there are many different TLRs, and it's difficult to knock all of them out, but they have only these 2 different adapter proteins, MyD88 or TRIF. So it's easier to knock out these 2 adapters. And then when I performed the acute liver failure model in these mice, I see that these cells, the activated cells from the liver, they look the same. They basically phenocopy what we see in MYC inhibition. So lack of TLR signaling and MYC inhibition looks the same. So there are the same populations in stellate cells and in endothelial cells. In Kupffer cells, it looks different because already at the steady state, the Kupffer cells look different. Probably their development is affected by lack of the TLR signaling. And when we look at the expression of the 77 gene signatures, the MYC signature, also in MYD88 TRIF knockout mice in presence of APAP, so in acute liver failure, there's no upregulation and no in neither of the populations that are resident. And also similarly to MYC inhibition, there's no infiltration of monocytes, but there is infiltration of neutrophils, shows that again, this is kind of specific to part of the phenotype. And after we established this, we wanted to see also if there is any effect in humans. And in mice, we observed not only these differences on the RNA level, but also we observe increase in the MYC protein levels with Western blotting. And in humans, we perform immunohistochemistries for MYC protein, and there's a huge upregulation of MYC in the nuclei of patients who have acute liver failure, or who had acute liver failure, and then they had liver transplants. And this work was done in collaboration with Dr. Shlomai from Tel Aviv Hospital. Yeah, so to sum up, I showed you that there's a common MYC regulated activation in stellate cells, endothelial cells, and Kupffer cells, and that it is regulated by the microbiome, and it affects infiltration of monocytes. And also, I didn't show you, but we also look into how the TLR and MYC are connected, and they are connected through MAP kinase pathway. So we established that using different inhibitors also in mouse model. And in the end, I would like to thank the lab I'm in now, the RNLinav lab. And yes, that's all.
Yeah, fantastic talk, Ola, and thank you for the memories. It was it was a good time. I was wondering, what is the like? Is the signaling for the ingress signal for the monocytes and neutrophils via is that directly via the stellate cells?
So stellate cells, it's all these actually cells express the chemokines. That the whole
Okay.
CCL2, it's kind of like a global.
And is that repressed? I mean, is the chemokine expression repressed through MYC pathway then?
So yes, so if I inhibit MYC,
okay.
then they are not there anymore.
Okay, makes sense.